A-level biology. A-level biology. And specifically, we are going to deal with monohybrid inheritance. Monohybrid inheritance. Say that this one is transmission of a single pair of contrasting expressions of a character. Inheritance or transmission of a single pair of contrasting expressions of a character. So, monohybrid inheritance lies under studies of Gregor, Mendel, Johan. The work of Gregor, Mendel, Johan. So how did he do his work? Experiment. Mendel used the garden peas. So can say he used the garden peas, a species called Pisum sativum. So he used the garden peas. He cross-pollinated a tall pea plant with a short pea plant. So what happened? Mendel saw or planted the seeds which resulted from the cross. So the seeds which were produced were sown or were planted. And so, what was the observation? Mendel observed that some seeds he observed that all the plants produced here were tall pea plants. Observation. All the pea plants were tall. All the pea plants were tall. Now from here, Mendel went further and then selfed the individuals of the F1 generation. In other words, he self-pollinated the organisms or individuals of the F1 generation. So that was step number 2. Step 2. Mendel self-pollinated the organisms or the plants of the F1 generation. So the plants which resulted from the first cross of the pure breeding plants, short pea plant and the tall pea plant, were called F1 generation. So you can say these were called F1 generation. F1 generation or filial 1 generation. So step number 2, Mendel self-pollinated the plants of the F1 generation. And so what happened here? Observation. Some pea plants were tall. Some pea plants were tall, while others were short. So when F1 plants were self-pollinated, then both tall pea plants and short pea plants were produced. Of course, when we shall be looking at the conclusion, what shall we say? That in the first generation, the offspring or progeny which were produced were heterozygotes. They had a recessive gene or a recessive allele in their genotype. It was masked or hidden by the dominant allele or the dominant gene. So Mendel, after doing that, summarized the results in a cross using suitable genetic symbols and crosses. So summary of the results. Summary of the results. Summary of the results. So we have what we call the F1 generation. F1 generation. So we have parental phenotype, the physical outlook of the parents. We have tall pea plant. We are crossing it with short pea plant. So physically, the parents are tall. One parent is tall and the other one is short. So we have parental genotype. Remember that the parents are diploid, that is 2N. They are diploid. They have 46 chromosomes. Their cells have 46 chromosomes. So this parent is homozygous for tallness, and the other one is homozygous for shortness. It is a dwarf plant. The genes undergo segregation. They segregate during meiosis. Meiosis occurs, leading to segregation of the gene or the genes, as shown here. We still maintain the sign for crossing. So here we shall have these recessive alleles separating, as shown on the board. So the individual alleles are carried in gametes, which are haploid, that is N. So each gamete can carry a single allele or a single gene after segregation or separation of the gene during meiosis. Next step will be random fertilization, that any allele of one gene pair can potentially and randomly fuse or combine with any other with any allele of the other gene. So here we shall have our crosses. We shall now cross as shown here. We are carrying out the crossing as observed on the board. So here we can write that this is random fertilization. Random fertilization. It is called random fertilization because there is no order, just as you've heard that any one allele of one gene can randomly combine or fuse with the allele of the other gene. So this one here, this cross, actually all of them will give us offspring with genotypes capital T and small t are shown here on the board. So this is F1 genotype. F1 genotype. Of course, the diploid condition has been restored, so it is 2N. So F1 phenotypes, how do they look like physically? Physical outlook, what we call F1 phenotype. All these offspring are tall. So we say all tall pea plants. All tall pea plants. All tall pea plants. So we said the next step now, we are going to represent step 2, where Mendel selfed the F1 individual. Selfing F1. Of course, that is F1 individual being crossed with an F1 individual, which gives us the F2 generation. F2 generation gives us the F2 generation. So we shall have parental phenotypes. Parental phenotypes. We have tall, the plants are tall, crossed with another tall pea plant. Tall pea plant crossed with another tall pea plant. So, parental genotypes, they are diploid or diploid, that is TT. They are now heterozygotes. Now heterozygotes are shown on the board because we are crossing the F1 individuals. The genes undergo segregation to form independent alleles, are shown on the board. Still also this gene will segregate to form independent alleles. This is double recessive. Independent alleles are shown on the board. The genes or alleles are carried in gametes which are haploid after segregation. Then, what happens thereafter is random fertilization. So we can now cross. We can now show our crosses as shown on the board. We can now cross. My cross is not sharp. We can now cross as shown here. Also, we shall cross this one here again with we shall cross. We have so far shown how one allele of one gene can cross with the alleles of the second gene. Then next will be this allele crossing with this one here. And also, my crosses are not clear. So, let me do it perfectly so that people can observe very well. The same, they undergo segregation. I showed here. I want to make perfect crosses. So this is T. So I can now cross perfectly. I showed here. Then this one will cross with this. Also with this one here. I shown on the board. So also this one happens during random fertilization, random fusion of gametes to form F2 with genotypes. This one will give us capital T, also capital T. This cross here gives us capital T small, also here capital T small. Then this one is double recessive, I showed you here. So we can call this F2 genotypes. F2 genotypes, that is 2N. Diploid. The diploid condition has been restored here. So we can also talk about F2 phenotypes. F2 phenotypes. We have these three plants. These ones here. They are tall pea plants. And then one is short pea plant. So now, when you observe the F2 generation, you can see or you can observe that individuals of the F2 generation are produced in a ratio of 3 tall pea plants to 1 short pea plant. That is the phenotypic ratio. Phenotypic ratio is 3 tall plants or pea plants, 3 tall pea plants to 1 short pea plant. So now you can observe that in this generation, the F2 generation, there is a character which has expressed, but it never expressed in the F1 generation. In the F1 generation, the recessive allele denoted by small t was hidden or was masked by the dominant allele or the dominant gene. But now, this allele has shown up in the F2 generation, as you can observe here. So, on crossing the heterozygous F1 individuals, then what happens is that the character can now show when two alleles which are recessive from either parents fuse at fertilization or during random fusion of gametes, then we produce some plants which are short. Of the plants which are tall, we can observe that some of them are homozygous dominant for tallness, while others are heterozygotes. They have the dominant gene and then the recessive gene, or a dominant allele and a recessive allele. So now here, what can we say? If we compute ratios from the cross That is the F2 generation of the cross. What do we observe here? We can see that homozygous individuals, you know that total ratio equals 4 plus 1, which is total ratio equals 3 plus 1, which is equal to 4. So it means homozygous individuals, these ones are dominant. Homozygous dominant individuals will be 1/4 times 100%. If we wanted to compute in terms of percentage, so this one will give us 25%, meaning that 25% of the offspring produced in the F2 generation are homozygous dominant for tallness. Then heterozygotes, these ones are 2. We shall take these genotypes here. We shall have, they are 2 out of 4 times 100. So this one gives us 50%. 50%. Then, lastly, homozygous recessive individuals. Homozygous recessive individuals will be also, it is just these ones here, 1/4 times 100, which also gives us 25%. So it means that out of those individuals of the second generation, the F2 generation, 25% of them are homozygous dominant. The other ones are heterozygotes. That is the hybrid group. So that is the summary of the results.